Oh, I also want to mention the Nobel Prize in Physics and Chemistry went actually to physicists, which is great. 6 of them, so half of the people on this thing, in fact maybe more, are physicists really. So these 3 people got it for the blue LED, which is extraordinarily important because you couldn't have a smartphone. You would have to have a 100-volt power supply for the light source for the screen of your smartphone until you got the blue LED, for example, also solid-state lighting and so on. And these guys got it for super resolution microscopy, and these guys got it for working out the map, how the brain tells you where you are. So how you're going to find your way back to the coffee or to the car park afterwards. The neurons, the place neurons, which tell you where you are now with respect to some framework. He worked out the place cells, I think, and these 2 worked out the cells that tell you the framework within which you're current places. And these other people did something probably useful, who knows. When we think about imaging, fundamentally, imaging in tissue, we need often to image deep in tissue, but most of the time we would really like to have cellular resolution. But in fact, that's not what we get. Usually what we get is with ultrasound, we see very deep, but we get 1 millimeter resolution. With MRI, we see very deep, we get 1 or 2 millimeter resolution. Also, PET is the same. Down at the other end, confocal microscopy, we can get subcellular resolution, wonderful resolution, better than 1 micron, but the depth we see is less than 1 millimeter, and the optical coherence tomography that I am going to say a little bit about, and Professor Pontiliano is going to say a lot about, is somewhere in the middle, where we see maybe 1 millimeter into the tissue, but we get almost cellular resolution while we do that. I used to show that picture, but I realized last year, or maybe the year before, that it's actually a little different. It turns out that all of these imaging techniques, even though they have vastly different fundamental principles, obey this rule where the axial resolution limit is 200 pixels. And very simply what that means is that if you want to image 30 millimeters within the tissue, 3 centimeters, then you get 3 centimeters divided by 200 is the best resolution that you can have of any imaging modality. And if you want to image 1 millimeter within the tissue, you get 1 millimeter divided by 200, 5 microns, as your best possible axial resolution. Adrian is probably going to defy me tomorrow by showing something better than that, but everything I have seen so far follows this rule. So if somebody tries to sell you an imaging technique for any kind of scattering medium like human tissue or animal tissue, and they say they can image very deep, but they say they are very high resolution, usually they're talking about 2 different things. So sometimes we can image very deep, but when we image very deep, we don't get this good resolution. In these other circumstances, we can get better resolution. So it's important to tell the difference between those, and this is especially true where we use laser-based technologies. But it's very curious that it holds up right across the level here. And the first time I talked about this was at a conference in Stockholm, and this guy, Stefan Hell, was there, and he came up afterwards to talk to me, and I said, this is great, I really want to know if this holds up for super resolution microscopy, because he is really the leader in that area. But of course, Stefan didn't want to talk about that at all. He was interested. He had just downloaded our app, and he wanted to know how this thing works, and so on, which we talked a little bit about, but in any case, we come back to this, and he said, the deepest we have been able to image in a mouse brain is 120 microns. And as far as I know, he said, the resolution at that point, axial resolution, now the resolution in depth, is 400 nanometers. So still pretty much holds up with this idea. So I think this is a quite interesting concept. Usually we would like to defy that and get beyond it because the changes that happen during things like cancer are happening at the nano level. So we'd really like to know what's going on at the nano level, but deep within living tissue. And by deep, I mean at least a millimeter, where there's a blood supply, there's a microcirculation that we've been talking about. Because this is real tissue, this is biology in its real 3D background. And most of what we know at the nano level, all of the techniques that work at the nano level happen just for superficial measurements. So atomic force microscopy, electron microscopy, where you coat the surface with gold and so on. All of this stuff we see maybe the first 10 microns or maybe 50 microns or so, but not more. We would really like to see what happens below that. And so we developed a technique which can at least get nano sensitivity, so we can see the changes in the structure at the nano level. And we published that last year in nanoscale. But now we have an exciting one in Nature Scientific Reports, which has just been accepted. It's not out yet, which talks about super resolution, going beyond the physical resolution of the system, using label-free techniques. So those guys got their Nobel Prize for label techniques using fluorophores. This is partly why they gave them the chemistry prize, because there's some chemistry in that. But you can't always do that, and that alters a little bit the biology as well. So it will be interesting to see what we can do with this. So I explained a little bit about OCT because I wanted to talk about what we can see in the microcirculation using this fantastic three-dimensional technique. It has a light source and it has this kind of Michelson interferometer, and we can build up images of the microcirculation, or of the tissue at least. So I want to talk a little bit about that. Usually in order to get this scan, you move a mirror by 1 millimeter, and that scans 1 millimeter into the tissue. These systems cost about 120,000 euro, from about 50 up to 120,000 euro for the clinical systems. Used a lot in the eye, but also now intravascularly for looking at plaque in the coronary artery. We are always interested in mobile and, I suppose, cheap and cheerful technologies that can hopefully solve problems for the 6.5 billion people in the world that will never access any instrument that costs anything remotely like 100,000 euro. So here is an idea to build a 10 euro or $10 OCT system, and it works on the idea pretty much of a CD-ROM or DVD drive, all more or less the same components, with this voice coil and so on, except and is essentially an OCT, a regular time domain OCT system, except that we put a partial mirror in here, and we still have this voice coil. So basically the same components except this partial mirror. And this partial mirror allows us to get many reflections from within the tissue. So the first reflection will build up a picture from very close, the first maybe 20 microns, if you move the mirror 20 microns. The next reflection will get you the next scan, and it will scan deeper and deeper and deeper into the tissue until you can build up a whole millimeter by moving your mirror only 20 microns. So why is that important? It's important because you can do it in a very small, compact device that costs very little money. It's very hard to make something move 1 millimeter many thousands of times per second. Okay? But to move 20 microns is not so difficult. Alright, so I'm going to maybe skip over that. And this allows us, for example, if you want to look at some curious thing on a patient's skin, you can point at the area and you can get a scan from within that area on what's happening at different depths. And usually with this kind of stuff, this melanoma, if that's what you're wondering about, you want to know how deep is that melanoma, right? Because if it has become invasive, it's gone into another stage, if it's gone into a deeper layer of the tissue. So we can see all these layers of the tissue. This work done by Joey Enfield and Ina in my lab. And you can see the sweat ducts here. You can see this fingerprint. So the secondary ridges, which are on the surface of your fingerprint, those small little things there. And there is a copy of them deeper down called the primary ridges. These are the original ones. And then the microcirculation actually follows the primary ridges, and the primary ridges follow the surface ridges. And not only that, these are the little loops that are coming to the surface. They look like dots because they're coming up, but we are cutting across them, right? And then the superficial papillary plexus that is feeding that is horizontal. That's going across, feeding these loops, and that follows also this pattern. And we think that because these sweat ducts, the microcirculation and everything is following the fingerprint at the surface, it becomes really difficult to defeat a fingerprint sensor that also sees the microcirculation. So it could be a much better solution than this solution that's integrated into the iPhone, which was defeated within 24 hours. Right? You cannot print something with flowing blood in 20 micron vessels with this pattern with any technology that I know of. And an even better way to image the microcirculation, as long as it's not your finger, is corrosion casting. Right? This is where you fill the vessels with some material which is opaque to X-ray, and you do an X-ray CT, I guess. And you corrode away all of the flesh around it, so you see this nice images. But you can see the pattern they are following this pattern on the surface. I want to talk about something really exciting I saw about 2 weeks ago at a conference I held in Ireland called BioPic, and one of the presenters, Patrice Mollard from Montpellier, showed some exciting results about a very hot topic at the moment on pericytes. And so these pericytes are the sites from the Greek, which means that they surround the vessel. So you have endothelial cells, then you have a basement membrane, and then you have these pericytes which surround the vessel. And the picture in our textbooks of how the microcirculation works talks about these pre-capillary sphincters, which say that these vessels are being controlled by sphincters at the beginning of the vessel. And this is how we've understood it for 50 years, maybe more than 50 years. But just now I have found that, or been told, that actually the pericytes all along these capillaries here are all acting, so the whole vessel is acting, the pericytes are acting to close these vessels entirely together. So this is actually fundamentally changing our understanding of the microcirculation. And I want to talk about, can I do this? So this is what we see when we look at the microcirculation. This is the type of thing that we see. So the cells going through larger vessels, and then these smaller vessels which have only one cell wide, and they're giving up their oxygen and taking carbon dioxide and other crap away from the tissue. And you can see the flow of blood from here to there within the tissue. And the way Patrice figured out that the whole thing is working is using something called optogenetics. So I want to go back to here. And this is just showing that actually living cells only exist within less than 100 microns of a blood supply, right? So this is within a tumor. It's a little bit larger, almost 100 microns but within normal tissue it should be of the order of 65 microns from the microcirculation. And because I did this quickly this morning, I borrowed some of these slides from somebody else, so just giving them credit. And so optogenetics is a combination of optics and genetics. So it has a light delivery method, and it has a genetic modification of the cells involved in it to make the cells respond to a particular type of light. The background to it is that our understanding comes actually from our understanding of how the eye works, because the eye works in the reverse way, where we have cells within the eye which are activated by light, and so they generate an electrical signal. And Edward Boyden very recently demonstrated this thing called channel rhodopsin, which is the molecule that causes that activation under light. Okay? So it causes neurons to fire essentially. And this has really just become hot in the last few years, but very, very hot topic. It works by introducing these optins into the DNA material, and this is transfected then into the cells, and you have a light delivery mechanism which causes them to fire when the light is shone on them. So basically it opens up some channels, ions flow from one side to the other, and you have a membrane potential, a voltage. For the physicists here, you have a voltage set up across the membrane of the cell. Okay? And that voltage being set up there is what causes, for example, my arm, my muscle to contract, to bring my arm up when my brain tells it that's what should happen. Okay? So I suppose I can't really go into the detail of this, but you're introducing the light. Most of this work is done in the brain because people want to be able to control neurons and understand which neurons do what within the brain. And so it will be really how we understand how the brain works. And here's an action potential. You have some stimulus. The membrane voltage is minus 70 volts normally. The action potential causes some sodium ions to leak out or leak in, I'm not sure which way around it goes, and causes the action. That causes a cascade of other ones to do the same thing, and you have what is known as an action potential, which causes the cells to do something, like muscle cells contracting. Okay, and there are various types of delivery mechanisms for this. You need to be able to get the light to everywhere within the brain or the tissue that you want to activate. And there are issues with getting the light in and sensitivity and so on. It's all based on this Nobel Prize winning work explaining how channels work within cells, and the idea that you have light activated opsins within the retina, which are essentially the detectors, the camera detectors within our sight system, translating the light into an electrical signal. Okay? So you have these rods and cone cells that are activated by light and send signals to the brain. One of the reasons that we're celebrating the Year of Light in 2015 is because of this extraordinary seminal work of Alhazen in 1015, and I suppose it's based on the teaching of Galen, which goes back even further. But he was already able to draw this a very interesting description of the eye, which is pretty much how we see it today. Okay, this is my much simpler explanation of optogenetics. Okay, and back to this slide here, which shows us again the pericytes around the surface. So what Patrice was able to do is cause these pericytes to be activated by light, and he did that using optogenetics. So he genetically modified the pericytes so that when he shone light on them, he could cause them to be activated, and if the light is not shone on them, they're not activated, and so on. And so he was able to show that the entire capillary was able to constrict, right? Not just these capillary sphincters at the beginning, which is interesting, and it will change the way we view the microcirculation. And he was even able to show that it is possible to cause one capillary to act in the reverse direction. So for example, this capillary here, by manipulating the pericytes on this vessel and this vessel, you could cause the blood to flow either that way or that way through that vessel. So you could actually change the direction of blood flow within vessel. And this happens naturally within the tissue, and we believe that it happens because some areas need more time to give out their the molecules, or to take in molecules, and this is partly done by making these vessels very small and very close interaction with the tissue. There's a very large surface area, even though it's I think Professor Barry said it was 5% of the output. It's actually 19,000 kilometers of capillaries versus a few meters of arteries and veins. So this is where all that interaction actually happens. Okay, so how do we image it in our lab? Well, we use this thing we call correlation mapping, OCT. And correlation mapping, OCT, works by looking at static tissue and seeing that that's the same from one frame to the next, and looking at the blood and seeing that that's different from one frame to the next. So this, if you do a correlation map, this will have high correlation always, and this will have low correlation always, because it's different from one frame to the next. And if you do that, you can build up these maps of the microcirculation. So this area here is where there was a low correlation. That bright spot means there's blood there. The dark spots, there's high correlation, which means there is no blood, there is no change from frame to frame, there is no movement in that tissue. Okay, let's skip maybe over this stuff. This is physically how we do it. We produce a kernel, and each kernel produces a correlation pixel. All right, and we mask out the tissue so that we just see where the movement is, and we get these kinds of images. So this is this area here within my forearm, and you can see all these small vessels, and we can section deep into the tissue, which is the thing we really wanted to be able to do from the start, and why we got interested in OCT for microcirculation. And as we go down through the vessels, we can see the tips of those rising loops, and then you can see the horizontal plexus, and so on, as you go down through the tissue, deeper and deeper. Okay, all right, and you can see the difference between healthy tissue, the border of a plaque, and there, here you can see the center of the plaque. This is psoriasis plaque. So you have all this confused blood supply, not this nice distribution of blood vessels that you should normally have. Okay. All right, and it's also useful for this kind of fingerprint detection. And in the nail fold is peculiar because it is one of the few places other than the eye you can see the microcirculation with a microscope. If you put a drop of oil on your nail fold plexus, the little piece of skin that goes over the nail, and put it under a microscope, you can actually see the blood flowing in that area under a normal microscope. So that's a useful thing that pretty much everybody can do. Here we see it in 3D using OCT. And by the way, the reason you can see in the nail fold, unlike everywhere else, is two things. One is the vessels are very close to the surface because they're above the nail, and they're going horizontally. so they're much easier to follow because they're going horizontally. You don't just see the tip close to the surface. Okay? And when we look at these pictures of microcirculation, here's one of the first videos we took in the forearm again, doing an occlusion, no flow, and then back to a lot of flow and hyperemia afterwards. And if we look at just one small capillary vessel, we can do it faster. I guess now we can do it faster anyway, but you can see that small vessel, how it comes back and is more active after the occlusion than before. And now we can see this reaction at all these different depths within the tissue. From 30 microns, where there is nothing happening because there is no blood there, to, well, in this case, 370 microns below the surface. You see there's a lot of flow there. Okay, and so yes, so I've spoken about most of those things, and I want to go on to talk about photoacoustics. And so we recently acquired 2 photoacoustic systems, one in the lab for animal work and one in the hospital for human work. And this is what we can see with humans. So these are some of the first human photoacoustic images. And photoacoustics is like ultrasound. It uses ultrasound. In fact, it listens to the sound coming back to the surface, but we stimulate the sound using light. Okay? So what we do is we put in a pulse of light. Maybe I'll explain that in a minute. We'll just show you the anatomy of this. So the grey is a normal ultrasound image, and the red is where there's a high absorption of the particular wavelength that we used. In this case, 860 nanometers. And you can see that that light is being absorbed more in the blood vessel than in the surrounding tissue. Not much more, but enough that we can see a difference. And so you can build up these pictures then of the microcirculation, and you can see what is universally true, that the higher the frequency of the ultrasound, the better the image resolution and the image quality, except that you lose the depth ability. Okay, so you can see much deeper with 15 megahertz, but you can see much better with 40 megahertz. Okay, if you use 800 nanometers as opposed to 1000 nanometers, you can see maybe slightly better. And we see the top and bottom of the vessels predominantly, because the sound goes radially out from the vessels. Okay. All right, and the last thing I wanted to talk about, I mentioned fluoroscopy, where we look at the coronary artery, and within the coronary artery we're usually trying to place a stent here. So this is a patient in our hospital. This is a view from inside the vessel. We get this from optical coherence tomography. So now we have the OCT system inside the vessel, and it's looking out at the walls. And the advantage of this is we can see what is the makeup of the walls. Is there a fibrous plaque or a lipid plaque or calcified plaque within that area? And we can also see the lumen of the artery, so we can see where there is a stenosis and how severe that stenosis is within the lumen. And currently, people use an extra catheter with a pressure sensor above and below the stenosis and calculate using the pressure above and below what is the fractional flow reserve. It's really a measure of how much your coronary artery is still able to produce a response when needed. And it's to do with the flow and how constricting the orifice, if you like, or the stenosis is. If you use the minimum lumen area, you can get a reasonable correlation with the fractional flow reserve. But actually the best, the most sensitive and specific response is from using this area within vessels that are less than 3 millimeters. So it just means that you can now use the OCT system without the extra catheter. And the extra catheter, putting an extra catheter in an already sick and compromised patient is a risk, let's say. And the last thing I wanted to say, which relates really to the microcirculation, is that we wanted to be able to see if not only that there's a plaque, but in this plaque, are there little blood vessels within that plaque? And this is very difficult to do because we're inside the coronary artery. It's near the heart is beating away, the coronary artery is moving with the heart, the lungs are going, everything is going within the system, and we want to see blood cells that are moving at maybe 1 millimeter per second within a small vessel in the wall, and that's very, very difficult. But we found that by using a second order correlation statistic, it was possible to separate this so-called static tissue, which is actually moving in this case in the coronary artery, and the actual flow. So we were able to separate this, and we just published in the last 6 months maybe this thing which shows us where these small vessels can be within the vasovas, or within the wall of another vessel. There is some microcirculation in the diseased state. So I went through a very kind of broad area, added even some extra stuff on optogenetics at the last moment, but hopefully you could get something from what I said. In summary, we have scattering and non-scattering tissues. We need to think, if we want a microcirculation imaging system, about what speed do we need to be able to image very fast, or we just need very nice pictures, still pictures. Are we interested in functional activity, flow or oxygen and so on? And are we interested in what's going on within the voxels at a resolution which is higher than the resolution of the voxels? And in all cases, the most important thing, as most of you are engineers here or applied physicists, is that your apparatus should be fit for purpose. Okay? So it doesn't have to be the fastest one, it just has to be fast enough. It doesn't have to be the highest resolution, it just has to have high enough resolution. Okay? It doesn't have to see, through the wall, it just has to see deep enough to see whatever it is you want to see. And with that, I want to finish thanking everybody who did the work in my lab, and thanking you